Okay, hello everyone. I wanted to go over a few more things. I don't know where our hour went. I think with our abstract reading it went a lot faster than what I was expecting. So I want to go back real quick. Lots of questions on the nuclear lamina, the nuclear envelope. The nuclear envelope, if I go back and look at, okay, there's gaps here. These are the nuclear pores. These are proteins. The nuclear lamina is kind of, if you laminate something, the nuclear lamina is like these fibrous proteins that are kind of forming this mesh on the inside, trying to keep everything together. And this seems to be continuous throughout the nucleus to keep everything kind of together in the nucleus. And this is all done by proteins. So that was the first thing I wanted to make sure to do. The second thing is the nucleolus was a question. And the nucleolus, and I think I misspoke myself, so I want to go back. The nucleolus is this mass of chromatin, mass of DNA. And its only job, what that DNA does, it's the site of ribosomal RNA, okay? And that in itself, its job is to produce ribosomes, okay? So it's a mass of chromatin, so it looks a little different. I don't know if it would look different to the naked eye, but it's a mass of chromatin that actually just makes the factories. So when the cell actually divides, that kind of disappears along with the rest of the... So what I'm saying is the chromatin that you have in your cells, actually part of it goes to making the actual ribosomes in the first place, which is pretty important because ribosomes are the site of protein synthesis. So it's just an extra mass, or it's part of the chromatin mass, but that's the area where proteins, the factories are being made. Sorry. Okay, so I want to go through a few things. Right now, as of anything that we talked about today, from class up to the nucleolus is on the quiz. So electron microscopes, surface area, volume, things like that. This stuff right now is not. I want to try to get ahead, so it's not going to be very long. But I want to try to get through as many of this stuff because I want to get through this chapter pretty quick. Ribosomes on the site of protein synthesis. These are the factories. So it takes place out in the cytosol, cytoplasm. Remember, cytoplasm is just a region, but out in the cytosol. And also on the outside of the endoplasmic reticulum. So that's what this would make this rough ER. And it can be bound in the nuclear envelope too. But these are all sites of protein synthesis. And if you remember back from the picture, there was a lot of ribosomes. So part of that whole idea is made up of ribosomal RNA. It makes proteins. So there's a lot of proteins being made. You remember that proteins are also enzymes. So here's some different, this is an electron transmission electron microscope showing you free ribosomes. And here's endoplasmic reticulum with ribosomes stuck on it. So when you're thinking about what you want your endoplasmic reticulum to be in your cell, you need to also make sure that you put some ribosomes on there so that you can distinguish between rough ER and smooth ER. Rough having ribosomes, smooth ER not having ribosomes. Rough ER versus smooth ER do have two different functions though too. And okay, on this page. So this is really important. I want you to kind of take note here. This whole word, there we go, the endomembrane system. Okay, the endomembrane system is talking about like the inner, these are components that are either contiguous or connected via transfer by vesicles. So this is all part of traffic, I guess is what I want to say. So the nuclear envelope, which is we already talked about. We just talked about endoplasmic reticulum, rough and smooth, Golgi, lysosomes, which we haven't talked about, vacuoles, and the plasma membrane is aka the cell membrane too. So this, remembering these is pretty important. Vesicles are just packages of stuff. Okay, so that's what we mean by that. The endoplasmic reticulum, that is really we call it the streets, pretty much of the cell. There's a lot of endoplasmic reticulum. It can be continuous. It can go from really the plasma membrane all the way into the nuclear membrane. And we have smooth ER, which we talked about, and rough ER, which is studded with ribosomes. Here's a great picture of it. So this can be very extensive. Seeing, you can see all the ribosomes. They do call like some of these folds cisternae, which is a great word because cisternae is actually the folds that are in the mitochondria. So, and here is one of those vesicles. So as it buds off, and again, things are being made in here. We'll talk about that in a second. So here's a great picture. Here's rough ER versus smooth ER versus rough ER. And there's an up close picture and the electron. So smooth ER synthesizes lipids, metabolizes carbohydrates, so makes carbohydrates, detoxifies. I think that's a good one, and stores calcium. And you might not think that that's a big deal, but storage of calcium is used in muscle contractions big time. So this is really important, and we talk about that a lot in physiology when we're looking at a muscle contraction. There has to be a big flux of calcium that actually comes out. So synthesizes lipids, metabolizes carbohydrates. So look at all these different functions that you can think about and represent by your cell model. You don't have to have all of these necessarily, but you do need to remember them, but pick one to represent for your cell model. Rough ER has ribosomes, and they secrete glycoproteins. So proteins covalently bonded, so glyco, so carbohydrates and proteins. They distribute vesicles, and these vesicles really have proteins around. So these are really releasing things, and then those things are going to go to the cell membrane and actually get released. So this is the membrane factory for the cell, the rough ER. I always say this is a transporter. The rough ER is like the street, so this is a transporter. That's the big word, transporter, to let things out. It's not, yes, it's a site for ribosomes, but that's not its function necessarily. That's just a site. It's got a lot of surface area, so it only makes sense that the ribosomes would attach to it, but its job is transport vesicles. The Golgi. I like to call the Golgi bodies or Golgi apparatus the UPS store. Okay, they are membranous sacs. Again, look at that word, cisternae, so folds or sacs. And the function, it takes the products of the ER, and also it does manufacture a few macromolecules, but it really sorts them. Sorts and packages. So you take this package to the UPS store, and the UPS store, if it's something fragile, put like bubble wrap around it and all that stuff. That's really the function of the Golgi. So that's, again, a membrane-bound structure. That's an endomembrane. And it's kind of interesting. It kind of is like pancakey. Like they look like pancakes, and so the vesicle will pass from one pancake to the next and get packaged with more stuff. See what I'm talking about? Like it'll go through here and then get packaged out, which is really, see all these little pancakes. And again, look at, isn't that amazing that you can see that now using a transmission electron microscope? And again, cisternae is just a fancy word for folds. There's a picture. Lysosomes, one of my favorite ones. I think it's one of the easiest ones to find a function to. A lysosome is a membranous sac. It can, so hydrolytic enzymes. It's full of enzymes, full of digestive enzymes that can break down macromolecules, which would include your fats, polysaccharides, nucleic acids. Works best in acidic environments inside the lysosome, which I thought was interesting. And you'll have to, there's different animations that I think you can see on your student section of the website. And so a cell can engulf. When you engulf something else, a lysosome has to break down. Actually, it fuses with the food vacuole and digests the molecule, so it actually breaks apart that membrane. And it also, the lysosomes are used to actually digest worn out parts of the cell itself. So it's kind of like the garbage disposal is kind of what I think, because you can put all kinds of things down the garbage disposal. And You can also, the lysosome would also be responsible for cell suicide. So if the cell needs to die, which some cells do need to die, and you do have cells dying, it can digest itself, which I thought was interesting. So actually, when you digest your own cells, it's called autophagy, which phagocytosis. So auto means self. So self phagocytosis. Remember, phagocytosis is just cell eating, which you had in biology. Here's some. You can see the lysosome and then the food vacuole that it comes in, so it can go and digest. Here's mitochondria and it digesting, so just kind of interesting. Peroxisome, we'll talk about that in a second because peroxisome sounds a lot like lysosome. And you can always, of course, slow these down. Vacuoles, again, an endomembrane. Plant cell or fungal cell may have several of these, and these are derived from the endoplasmic reticulum and the Golgi apparatus, so there can be vacuoles. There are food vacuoles, contractile vacuoles. One particular organism that has a contractile vacuole was the paramecium that we looked at your freshman year. That's the one that has a cilia, kind of looks like a foot. And central vacuoles are found in mature plants, too. So we can have different types of vacuoles. There's the central vacuole of a plant cell. It has a lot of food storage. And that was all of the endomembrane system. Again, complex and dynamic player in the internal part of the cell. And you can look at, there's the Golgi. You don't have to, the cis trans means beginning to end, so you don't really have to remember the difference between those, but here's the vesicle entering and then leaving, so as it gets packaged and packaged and packaged. And there it is, fusing with the cell membrane and then releasing the products, which this, by the way, would take energy, most definitely, for the cell to do this. And I think we'll stop for right there. We'll talk about mitochondria and chloroplast. Yeah, we'll talk about those tomorrow. Okay? Thanks for listening, and hopefully we'll jump ahead a little bit. We'll talk about these tomorrow.